Today I'm going to present a couple of projects. I thought they were kind of nicely related. One is a much earlier project I did in my master's. It's a bit more of a hypothesis, so less of a complete project. And that's looking at the morphogenesis of the gastrovascular system in Aurelia aurita. And the second project that I did in my PhD is looking at animal cap elongation, so axis extension. And so the second one is looking at how two types of cells or cell populations organize within each other in order to create order from disorder, right? Okay. Uh, so, okay, so to start with, I thought to frame these kind of the projects that I'm going to show you is a paper that Dennis Noble wrote. It's called a It's looking at a theory of biological relativity. So he posits that there's no privileged level of causation. So usually we think of causality as tending to go from the genes all the way up to the organism. And instead, he was thinking about how higher levels of organization um, impact on lower levels of organization. So this is probably nothing new in this lab. And he came across this idea because when he was doing earlier work on cardiac oscillations, so he found that when he modeled just the proteins in the system, he found that the oscillations would go to a steady state, and unless he added the boundary condition of the entire cell itself, he would lose those oscillations. So he found it was really necessary to have that outer boundary condition from a different level of organization. And so patterns of activation can really come from kind of any scale. Um, and saying that, so in this project, so the hypothesis is actually in the jellyfish that the swimming contractions, so the short-term deformations, are actually patterning and guiding the gastrovascular patterning the of the canal system. So you have these kind of quasi-fractal canal systems that grow from the rim. And so this is really nice. You have these two kind of different scales, one's in terms of time and the other one in terms of the behavior, the actual swimming behavior of the jellyfish is guiding morphogenesis, which is kind of unintuitive in a way. And to tackle this question, we kind of have to understand how canals grow. Yeah, so feel free to ask any questions. I'm one of those ones that prefers people to just jump in, so go ahead. Get this first trifurcation. So it's this middle canal that grows first, and then you have the first two canals that grow and connect and join the youngest canal. And as the jellyfish grows, more canals start to grow from the rim and join. So it's kind of a fractal pattern in reverse, right? So usually branching patterns happen the other way around, but here it's kind of growing that just has grown. And so you get this kind of fractal pattern that keeps growing as the jellyfish evolves, right? So this is a continuous process because the jellyfish, there's no limit to how many canals will start growing or what the fractal pattern will do. Preset pattern or end point to this process until the jellyfish. So to think about how the canals might choose where to grow, we need to think about three different sort of main parts. So one is the canal system itself. So the canal system grows into the endoderm layer, as you can see from this diagram. So you have an endodermal sheet, and the canals kind of grow into that endodermal sheet and differentiate into canal cells. Sandwiching the canal is you have elastic gel. So it's kind of a gel that contains ECM, so loose ECM. And the third most important interacting part for us is the muscles. So the muscles run circular around the bottom of the and kind of these are the most important players in terms of thinking on the time scale of a contraction of a jellyfish. And so here again, you see the canal growing into the endoderm, and you can and here is an image of that canal growth from the rim. And so our hypothesis is that cells proliferate along lines of tension. All right, so this has been seen in many, many different systems. So one example is with the chick gut. So in this experiment, a small mass is attached to the intestine in a culture medium. And the larger the mass, the more growth you get, right? Because the cells are responding to these tensile forces. A second one is a really old experiment that I quite like. Uh, it's in it's in chick. So if you take the blastoderm with the vitelline membrane, usually people um, extract it and put it on a circular ring. Instead, if you put it on a triangular ring, the cells will feel the lines of tension, which will build up obviously at the points of the triangle, and you end up with a triangular shaped blastoderm. And so if this mechanism is at play in our system, so what I want to do is try and look at kind of where the tensions might be within the system. So first we want to identify a tensile stretch that's taking the canals to the center of the jellyfish, so an orthogonal tension. And secondly, as I said, the canals, they make decisions to join with other canals. So we kind of need a radial tension or something, an attractor that's pulling the canals into other canals. So these are the two that I'm going to think about and present to you today. So I'll just present mechanisms 1 and 2, so the mechanisms that guide cells to the center, and then also a kind of turning mechanism and suggest something for that. And I'll leave the third one for the sake of time. So how we do this is place the jellyfish inside a Petri dish and we film it with a high-speed camera. We get these beautiful images like this. Then we use PIV, so particle image velocimetry, to look at how the tissue is deforming over time. And this gives us an indication of how the tensions are in the tissues. Okay, so first I'll present a mechanism of how the canals get driven to the center. So this involves two parts mainly. So the mesoglia, so this sort of viscoelastic gel that I talked about earlier, and the muscle layer. And so here I make a crude measurement of strain in 2D as the jellyfish contracts. So if we just measure the mouth part, which is in blue in A, as the bell is relaxed, and then as it's contracted. And we see that the mouth actually, upon the contraction, the mouth is actually contracting and the endodermal part is stretching. So here we can already see that as the jellyfish contracts, you're stretching that endodermal layer continuously and continuously over time. And it's always consistently stretching the canals. And you can see this more closely if we take just a section of the jellyfish. So this is a quarter of a jellyfish. And you can see as we do a close-up that the cells or the tissue is being stretched towards the center. And this is because you have the viscoelastic gel that's shaped like a wedge, right? And as the muscle cells contract, it's actually you have a conservation of mass and as the cells contract, it stretches the whole system in the other way. And so I've taken the same sample and left it overnight. So you kind of degrade the mesoglia part. And so what you do is you thin out the mesoglia. And as the jellyfish contracts and you have less mesoglia, you actually just get this buckling behavior. So you don't no longer get this stretch. So it really shows that the stretch that might guide the canals is really a coupling between these two materials, having muscles contracting and also having this thick viscoelastic gel wedge shape. Yeah, so that's just summarizing that. So the second mechanism I want to talk about is a turning mechanism. So this really relies on the canals and the muscle cell layer. So this came from like just observing as muscles twitch. So in this video, you can see the muscles twitch. You can see how the connection between the canals that are actually being moved at the same time. So the muscles are really affecting how the canals are just being pulled around. And if you just take a little pincer and just take just the very top layer of the muscles and pull it, you can see quite clearly that the canals even further away are being affected by that small pull. And so they're kind of dynamically attached through this muscle mesh, the canals. And so again, we do the same kind of process. We take an area of crop an area of interest. So in this video, we have an older canal. So it's this thicker canal that you can see, and we have a small, younger canal growing away from it. And we use PIV analysis also to look at tissue deformation. And interestingly, we found we can create a deformation field based on that contraction. And if you can make out, you can see that the canals tend to compress more than the surrounding mesoglia. And actually, in fact, the younger the canal is, the more it will contract. All right. So if we look at the percentages of the area, we see that we're contracting much more. Younger canals contract much more than older canals. This is probably because you have a growth of ECM as it's getting older and stabilizing. But this creates a kind of interesting dynamic in terms of creating different tensions. And so in this deformation field, you can see between the two canals, you can see lines that are pulling between the two canals. And so the theory would be, so if you have three canals, one is the youngest growing in the center, and you have two canals on either side. So 3 is stiffer than 2, and 2 is stiffer than 1. And then you have a contraction. So this will create tension in the surrounding mesoglia. As the younger canals contract more, you would see that there would be a bias towards the younger canal because you create larger tensions in the mesoglia, in the surrounding tissues. And we find this creates a kind of feedback where the other canal will start to grow towards it. So we think that this is the turning mechanism that defines where you'll either or a younger canal. So these are the kind of, we think of the rules that are governing how the canals make choices, first to move to go to the center and then to make a choice to turn.
Can I ask a quick question?
See, yeah,
Yeah, so in this case,
go ahead.
maybe I missed something, but in this case, wouldn't it turn towards E3, which is the stronger contractor? I'm not sure I understand why it
Well,
bends to E2.
so E2 is because E2 will contracts more, so there's more tension between the two. So E2 and E1 will contract, and that will create a higher tension between the two
Okay.
than it would do with E3.
Okay.
Yeah, does that make sense?
I thought that in your little diagram on the left of the screen, you're saying E3 is greater than E2. So I would have anticipated the difference between E3 and E1 would be larger. No?
It's stiffer. So it's it depends on because they can contract more.
Mm,
So because E3 can contract less, it's pulling less on the actual surrounding material.
I see. Okay. Gotcha.
Right. Yeah, so it's like, yeah, I think intuitively you would think everything will grow towards the stiffer one, but actually as it's how it pulls the surrounding material. So if you can contract more, then you're actually pulling harder on that material than a stiffer canal that can't uh, pull the material as hard
Got it.
or create as much. And you create this like canals go to even meet each other.
Got it. Thank you.
And so it's a, yeah, does it make sense?
Yes.
Okay. Yeah. So there's a really, hmm? Yeah. So to summarize, canals that they're in this dynamic, growing system that's changing all the time. And it's responding to a kind of tensional field that is expressed when the jellyfish contracts as it swims. So this tensional field has younger canals that become like attractors as they're more pliant, drawing even younger canals towards them. So that's the idea. So you have, and they're directing lines of growth. So these are the rules that we think are directing, governing the system of how the canals will grow, how they decide to join with other canals, um, and create this kind of dynamic system that's ongoing. And as I said, it's really interesting because the jellyfish is this is an interesting system because it's where the organism is actually the organismal behavior is actually impacting on morphogenesis. And because the jellyfish is constantly in growth, you kind of have this system that's ongoing that doesn't need much maintenance in terms of like a pre-patterning, like it can go on eternally and it's very dynamic and no jellyfish gastrovascular system looks the same, right? It's a very dynamic pattern that happens um, within different timescales. Okay. Um, and so the second presentation is the one is a project that I've done in my PhD, and that's kind of looking how epithelial boundaries are actually organizing cell alignments and morphogenesis. Um, so here's five or some examples of embryogenesis. So embryogenesis is beautiful, as you all know, self-building, ever complexifying but modular system. So starting from a single cell that divides, differentiates, creates different tissues, they interact and can build a multiplicity of tissues that stabilize over time. And we kind of want to know how these morphological decisions are taking place. So even though there's vast differences in terms of starting geometry, size, timing, there are some fundamental similarities. And in the examples I'm showing you, sort of creating the anterior posterior axis is kind of one of those morphological motifs. And in all of these systems, they do it in many different ways. They get to the same kind of sort of pattern in a multiplicity of different ways. But the basic motif is that you will contract in one direction and extend in the other. So this is the example in Xenopus at different stages. So I'm sure you've all seen these gastrulation videos, but this is a view from the vegetal pole of Xenopus. So this shows the involution of the outer tissues through the blastopore lip, which is the process of gastrulation. So you have the tissues are deforming and converging and also extending, and then you also see the process of neurulation in this video. And as the Xenopus embryo is pretty opaque, we can have a look at it from the cross section. So this is another cross-section view. So you have the blastopore lip, the extension of the axis that you can see on the right-hand side, and the formation of the gut. So if you can compare the two, it kind of looks a bit like this. And it's long been discovered that the cells that are actually driving this process or convergent extension are the mesenchymal mesodermal cells. So like a long time ago, people noticed that all these cells started to align perpendicular to the axis of extension, and they saw the behavior of the cells, they start to crawl in between each other. And this is kind of it kind of creates this behavior where you kind of the cells start to stack up, and this is called convergent extension. And so what's important is that you need the cells, there needs to be an orientational order with respect to the axis and directed movements. And that's how you get this kind of basic morphological motif. And the two ways that we can do that at the molecular level, it's been shown that cells use anisotropic sort of pulling on their neighbors. And at the same time, they have this contraction mode, which is junction contractions in an anisotropic direction, which also coordinates this behavior. So you have both of these happening at the same time. And yet, Holt-Freder a while ago, he took out these cells, so these mesodermal mesenchymal cells, and he rearranged them and put them back together, and he found that they couldn't elongate. So he kind of said that there's something missing. There's something missing that's within the embryo itself that's not a property of these populations itself that are creating this kind of ordered cell orientation and this ordered behavior. And you can kind of repeat this experiment. So here I've done it with not with the mesodermal cells, but with animal cap explants. So you can take the mesenchymal cells and add activin. And when you do that, so these cells should be in a mesodermal fate, but they don't elongate. So they fail to find some kind of uh, morphological or like orientational coherency in order to propagate the elongation or make a decision. And when you add back the epithelium, you can kind of rescue this behavior. So no one really understood what the relationship is between these two cells, and that's kind of what I'm going to be looking at today. So how does the interaction between these two populations of cells enable elongation? And so one of the hypotheses was that the epithelium was just passive, right? It was just lowering the interfacial surface tension that would allow the mesenchymal cells to um, sort of get over the energy barrier they would need in order to elongate, right? And it was just creating a support. And they did this, Niedermeyer and Winkelbauer did this beautiful experiment where they took those mesenchymal cells and they made a kind of sausage. And they found that that sausage just rounded up as would a liquid. And instead, if you do the same thing with the sausage and now wrap an epithelium around it, you kind of maintain that shape. So with this experiment, they were trying to prove that, yeah, the epithelium is just helping the mesenchymal cells stay in that shape. And this another beautiful experiment by James Green shows that there is one condition in which you can get just the mesenchymal cells to elongate, but you have to start from a disc. So this is a video that he made. So if you start from a disc, somehow the cells are aligned along the boundary of that disc, and that is kind of remembered. And all you do is get this waist constriction that actually drives this elongation that you don't usually get. So showing that the starting geometry is really important in order to get the behavior that you want to see. And so he showed that there's a peripheral alignment zone. And yeah, and it also speaks to, the number of ways that cells can adapt in order to get to what they need to get to, right? And there's not necessarily doing exactly that, but like, yeah, you always find a multiplicity of ways that cells can get to the same morphological outcome. Yeah, and so to tackle this question, so I'm using a minimal model of axis elongation. So Sims and Smith, they identified that if you stimulated the animal cap explant and added activin at a certain concentration, you can push the cells into a mesodermal fate, and they will indeed elongate and make these kind of dumbbell shapes. And so this system kind of is really nice in a way, and I guess that you're really familiar with the system, but it really speaks to the modularity of morphogenesis. So I think it's really interesting that they can take these two population of cells and replicate something that's happening in the embryo, yet at a different geometry, right? So they adapt to a different geometry and they can still create these elongated shapes. And you can say we only need these two cell types in order, these minimal amount of cells in order to get that kind of patterning that we see. Um, and so here you see that with activin, you have these dorsomesodermal structures over time. And if you don't, you get sort of atypical epidermis or xenobots. Yeah, and what's really interesting about the system is that because there is no initial pre-patterning, you can have a little bit more control over the system. You can take it apart, you can put it back together, and you can do that free from the constraints. So you're able to ask different questions of the different tissues to see how they're interacting. And so this is the process that we make the explants. So we cut the animal cap at stage 9. There's an epithelium and 2 or 3 layers of mesenchymal cells. And these guys will always round up. And I kind of call this process synthetic epiboly because the epithelium does pretty much the same process as the whole embryo does. The epithelium will always engulf the mesenchymal cells, and at the same time, you always get this tissue boundary. And then after some hours, the tissues will they converge at some point and then they will begin to diverge again and elongate. And so this is kind of what it looks like. So what you're seeing is that the brighter cells are the mesenchymal cells, and the darker cells are the pigmented epithelial cells. And one thing that you'll notice is that explants always elongate perpendicular to the boundary between the tissues. So that was kind of something that was like very obvious from the very beginning. Okay, there's always one direction in which elongation happens. We also get this kind of contraction before elongation happens at the tissue boundary. And so the first thing we wanted to do is look at the sort of coarse-grained tissue deformation to see if there was any sort of if there was anything obvious in the tissue deformation that we could see. So we do this by embedding them in methyl cellulose, a bunch of them to stop them rolling around. We take a time lapse, we use PIV again, and from that we can look at tissue deformation by creating an advected grid that will follow, that will move with the vectors of each time point so that we can get the coarse grain look of how it's deforming. Yeah, so here is a chemograph of that process of an explant. So in red is the mesenchymal cells. And if we look at the tissue deformation and create the same kind of chemograph, what we find, so if you look at the purple arrow, is that the majority of elongation is actually happening from a really small region of that explant. It's happening right at the boundary between the two tissues. And if we look at the strain, you see that it's really just a small area that's creating most of the elongation. So this was kind of surprising. So it shows that elongation is really localized. And this is kind of, we guess, how you get those kind of dumbbell shapes. And at the same time, you don't see any elongation in any of the mesenchymal cells that are protruding um, from the explant. And when we looked just following cells, we see that uh, you know, the epithelial cells aren't passively being stretched, but they're actually intercalating, right? And so this is kind of the cartoon of what we see just looking at tissue deformation. So we see a thin bound that's actually you have this contraction and then everything starts to elongate from the boundary. you have a kind of it's a yeah a kind of waist constriction. And so the next thing we wanted to do is test all these tissues in isolation to see how they respond to activin. Uh, so so first we have the coupled tissues, so both tissues together, then the isolated mesenchyme, and then the isolated epithelium, which you can do by kind of at a certain point, it becomes easier to just remove just the top epithelial layer. So we also have done experiments that I'm not showing today where we do mix and match between an epithelium that has activin and a mesenchymal cells that don't and put them in different combinations to see what happens. Um, and so here you see the coupled tissues, you get this really nice, beautiful elongation. And no surprise when you have the isolated mesenchyme, which are known to really drive elongation in Xenopus, um, we don't get any elongation. And surprisingly, when we have just the isolated epithelium can just elongates by itself. So it does it in a really like unpredictable way, right? So there's not the same kind of order that you have when you have coupled tissues. So sometimes you get multiple poles, sometimes they don't elongate, and sometimes they elongate. And so what's really interesting, there seems to be sort of tissue coupling actually creates this morphologically stable, elongated form. And it's sort of, we think that there's something, there's a cooperativity between the two tissues that actually stabilizes morphogenesis. And so the first thing, so
Can
we
I ask
wanted
a
to
quick question on your previous? With the
go ahead.
isolated epithelium, where does the extension happen?
Where does it happen?
Yeah, does it happen all over? Because you mentioned that when you couple the tissue, you have a thin layer of cells that are proliferating, and that's where the elongation happens.
Right. Yeah. So when you map tissue deformation to isolated epithelium, you can have like so it's not over the whole thing. You do get an isolated region or two isolated regions. So in fact, this one that I'm showing you, I think if you look at the tissue deformation map, you would get two poles. So it's actually like you're getting elongation that's concentrating in two different parts. So it's this is kind of by chance, right? So I think it's kind of like opportunistic in terms of it depends on the starting geometry of the epithelium itself. So that can really vary quite a lot.
Mhm. Okay, cool. Thanks.
No worries. Uh, so the next thing we wanted to look at was cell orientational order in both in all of these kind of uh, um, samples. So both when they're isolated and when they're coupled. So we use a nematic order that to look at both cell elongation and cell alignment. And I should say in cell alignment, you get negative numbers. So the way the quantification works, that if you're in negative numbers, that means the cells are elongated perpendicular to the axis of elongation. And so we found that in the coupled epithelium and the isolated epithelium, so we take apart just one part that is elongated, so using those tissue deformation maps, so specifically a part that's elongated, and we compare both cell elongation, which how elongated cells are, and how aligned they are. And we find that there's pretty much no difference between the two, which means that sort of sort of this behavior is kind of intrinsic to the epithelium. Um, on the other hand, when we look at the mesenchymal cells that are inside the epithelium in this top example, we find that they're really, really highly elongated and highly aligned. Whereas if you have an isolated mesenchyme, there you get a similar elongation score. So the cells are trying to elongate, but yet they have nothing. They have significantly less orientational order. And so what this kind of says is that the mesenchymal cells are obviously responding somehow to the epithelium. So they need the epithelium in order to obtain that sort of orientational order that we know that is important for convergent extension. The next question we had was, how does cell orientation emerge? So we wanted to look, we wanted to see at early time points where orientation was emerging first. So for this, we're looking at the apical surface of the epithelium, the basal surface of the same cells of the epithelium, and then also in the mesenchymal cells. And we're doing this at these two time points. So this early time point before elongation happens, and then also as I told you at tissue contraction. So before we start to elongate, we tend to get these like contractions that happen at the tissue boundary. And so what we find is that actually cells don't look so ordered. So we look at this nematic order Q. So cells are not very well ordered on the apical surface of the epithelium. If we go down into the explant, we find that they're actually the basal surface of those same epithelial cells have started to align already. So alignment is already happening pretty early on before, so that orientation is starting to propagate even before you get any elongation. And the inner mesenchymal cells are a little still, even though cells are elongated, are pretty disordered. And at the second time point, we find that now Uh, still, the apical surface is not very ordered in terms of orientation, but we have high perpendicular orientation of the basal epithelial cells. And now the inner mesenchymal cells are starting to follow the epithelial cells. So you have this kind of propagation of orientational order that goes from the basal side of the epithelium to the mesenchymal cell. So it's propagating from a 2D surface into the 3D mass. And this is kind of all everyone is getting organized before elongation happens. And as a comparison, if you look at the outer mesenchymal cells, you see that they're fairly disordered. So there's some kind of inflammation that's happening between the epithelium and those cells internally, the internal mesenchymal cells. And so just to summarize, the propagation of cell alignment originates in the basal epithelium. And from this, we can kind of guess that it's a crawling mechanism that might be originating where the alignment is originating. So before I told you that you have these two methods of aligning. One can be from the apical surface or the basal surface of these cells. So we think that there's a crawling mechanism in the epithelium. It's then propagated down to the mesenchymal cells. And as I said, you have this propagation from the surface to the bulk of the tissue. And so the next kind of question we asked was, so what directional cues sets the axis of elongation? So this might be really obvious by now, but so we said earlier that elongation is always perpendicular to the tissue boundary. We also find that the epithelial cells always orient parallel to that tissue boundary, and that extension in this case tends to be localized to that tissue boundary. And so we hypothesized that it's actually the epithelial boundary that might be deciding the orientation of our explant. So to test this, we did these kind of fun experiments where you take the explant and you just chop it into either 2 or 3 pieces and put them back together and let them heal. So in this case, the mesenchymal cells will heal, but the epithelial cells, they stay kind of separate. So you have what we call these 2 tissue and 3 tissue sandwiches. And what we find when we do that is that you can either have 2 poles or 3 poles, depending on the fragmentation of the epithelial boundary. So nothing else has been changed except for just the fragmentation of the epithelial boundary. And it will direct you to give you any number of poles that you want. And so, yeah, tissue boundaries, like we find that they really organize morphogenesis. And we find that the fragmentation of the initial explant creates more boundaries, so you get more poles. And the cellular alignment information propagates from the epithelium to the mesenchymal cells. Um, yeah, and just as some concluding remarks on sort of both of the projects that I've just showed you. What's really interesting is that both of these processes don't need any kind of pre-patterning. And it's kind of not to say that pre-patterning isn't important, but I think that there's a certain amount of flexibility that when you respond in this way, so obviously the organism sets up these conditions that allow for the flexibility. And that means that in each system, we're reading, or the cells are reading the substrate on the fly. And I think these kind of systems might add to when we talk about robustness and flexibility. I think that the capacity for some cells to be able to do that is probably adds to that robustness that we often talk about in embryogenesis. The fact that you can constantly, through mechanics, read and adjust as it goes. So in the animal cap explants, it adjusts to a certain different geometry than it would be in the embryo itself. It can still do the same kinds of behaviors, but it can easily just adjust to a different geometry. And if it's following just simple rules, it has that possibility. Yeah. And with that, I would like to thank my lab, my supervisors, and everyone else that helped me work on the project. And I'd like to thank you all for listening. Yeah.
Excellent. Thank you so much. Anybody have questions, comments?
Yeah, I have a question if
Yeah.
Go for it.
so this seems like a really cool way of making kind of non-spherical shapes. So often you see people do organoid stuff, and just because of, sort of basic oil and water physics, things tend towards spheres. So I wonder if you were to take an organ primordia, let's say like an eye primordia or a heart primordia or something that will normally tend towards being a sphere, putting it into one of these kind of converging, extending organoids, do you think that you could make kind of variant organs, basically an eye that is elongated as opposed to spherical?
Right. I mean, I guess I don't think it's trivial. So like I think I mean I just mean in the sense that I say you don't need any pre-patterning, but
Mhm.
like the starting conditions are set up, right? Those cells
Mhm.
have those kinds of behaviors and those cells will behave in those kind of ways. And that's particular to those cells that aren't particular to, say, the eye primordial cells.
Sure. So
So,
I'm thinking that basically you have the ratio is like 10 to 1, right? So basically you have the system so that most of the mechanics and stuff are, the way that your systems are behaving, and just put, whatever the minimum amount of, say, I primordia, not so that it will contribute mechanically, but basically it will get deformed
I
by the
see what
movement
you
of
mean. All right, right,
right?
right. Okay. So to create like a kind of hybrid between
Exactly,
an
exactly.
an extending
Yeah.
yeah,
So
I
you
mean
take
yeah, I mean I guess it could be tried definitely. That would be interesting. A kind of fragmented eye across a extending body. Yeah.
cool. Thanks.
Yeah. Mm.
I've kind of a related, at least I think it's related question. Do you have any thoughts on structures that are very flat, like tadpole tails, planaria? Do you have any thoughts on how they stay so flat? Because to make something that's flat like this, you really have to coordinate, right? To make sure nothing grows in the third dimension, nobody bends. Like it seems like a difficult problem. Do you think it's a difficult problem? And what do you think are the mechanisms for growing something very long but very, very flat and straight?
Yeah. So I think that's true. Yeah. No, it does seem like a difficult problem, but I think yeah, that depends on the properties of um Yeah, no, that's an incredibly difficult problem.
Yeah.
Flat and straight, yeah. Yeah, so actually Matthias Merkel, one of my he's a theoretician who's kind of working on these kinds of problems. And working like how like obviously like when you have something like convergent extension, how do you stop the system just going into chaos as you would, like at some point something starts to bend and you start getting like, small perturbations might start, and you kind of see it, and you kind of see it in our system. So when you're free from other constraints of the embryo, these elongating shapes, they start curling, they start going all over the place, right? There's way more freedom. There's way more freedom that they have and the constraints in the embryo stop all of that. Yeah. So I guess that there are other structures that will help to preserve, like ECM and kind of, I don't know, there must, yeah. But in these things you see with the degrees of freedom they don't have that capacity to just keep going. So any kind of small perturbation, if you start to get a curve, that will just absolutely just continue curving and propagating. So any errors are not really able to be corrected if you don't have these other constraints. But yeah, saying that, like, I don't know exactly what the constraint should be for getting something long and flat. Yeah.
I have a couple of questions.
Yeah.
The first one would be, so in this context, does the proportion of the cells matter? As in, I'm assuming that here they look like about 50-50 epithelial versus mesenchymal. So would you be generating specific shapes if the proportions were different? And second question is, where then is the force coming from? Is it still coming from the mesenchyme, or because it seems like from your work that the mesenchyme can't do it by itself, and it's some part of the epithelium that starts the process. But is that like a
So,
duopoly
so
where?
both, so
Okay,
both tissues
okay,
are creating forces, right? So,
okay,
I mean, it's obviously shown that the mesenchymal cells do drive this, but as I showed you in the experiment, the epithelium will elongate by itself, so it has its own internal forces that are doing it as well. So it was not really known. I guess people just thought that epithelium was just a passive
gotcha.
cover, right? But actually, they're just these sort of boundaries and just become these kind of organizers, and they're not your kind of typical organizers. They emerge through sort of this coupling, right?
Right, right. And what would
Mm.
happen if there was only 3 cell layer thick epithelium on top of the mesenchyme? How the interaction would be? Do you think it would still be the same, or would the proportions of the cells change the kind of interaction?
Oh,
And
if you had just three layer.
yeah, I'm basically trying to understand if the number of or the number of cells or the proportion of epithelium versus mesenchyme has any effect on what the outcome will be.
Um, so I would say the more mesenchymal cells you have internally, uh, um, I mean, you get, I think the more cells you have internally, the more it sort of also helps the elongation to stay straight.
Okay. Okay.
So that's the kind of morphological differences you would have, and there's experiments that I haven't shown where, so we tried to see if we could get the epithelium to do exactly what it does, but without the mesenchymal cells.
Mm.
So we wanted to recreate just a boundary condition um, without the cells. So we attached it to a bead, and it creates a boundary condition where you just have the epithelium. And we found when you do that, you get a really nice you sort of guide morphogenesis, or morphogenesis is being canalized if you create a boundary,
Gotcha.
right?
And you think that happens best if the mesenchymal cells are completely covered by the epithelium? Not, it won't happen if they are side by side attached to each other. Is that correct?
No,
Or
it can
okay,
happen like that as well.
I
Yeah,
see. Okay.
it creates the same thing. So sometimes we would look. So I told you we were creating these mix and match where we would put the epithelium inactivin and the mesenchymal cells not. And yeah, you can create these kind of boundary conditions and it will still do the same thing. And sometimes it will elongate with the mesenchymal cells inside, and sometimes it would just elongate by itself.
Cool. Cool.
Yeah.
Thank you.
Just a couple of questions. Really interesting talk, by the way.
Thanks.
Do epithelial cells behave in this way when they're paired up with other cell types?
Ah, you mean like when there's like goblet cells and other cells
Yeah,
and
if you just don't use mesenchymal cells and you just put, I don't know, muscle cells or whatever, right? And would you see this sort of pattern as well? Is that, I guess what I'm trying to get to is it the boundary that's really crucial for this process to happen, or the specific interaction between the mesenchymal cells and the
oh,
epithelial
both,
cells?
both totally.
Okay.
Both
Um,
totally, because I mean, it depends, obviously I didn't go into the molecular sort of part of it, has to do with adhesion molecules, which is obviously very important in terms of like one, if you're but in one instance, if adhesion is too strong between these two layers, it would be really difficult to elongate. So you have to have a kind of a right balance of molecules that really are specific. So I made it very simple, but actually the molecular side of it is quite complex on fit. That's what I
uh,
said. Like it's you can read it in this simple way, but everything is set up just so that it allows certain possibilities because you've set the system up and it doesn't need any other sort of pre-patterning because you just set it up just right that it can do it by itself. And um I think that that's a kind of interesting, yeah.
mhm. And the other question I had was, this might be silly. Oh, just a clarifying question is, did you use two layers of epithelial cells? Or just, did I get that right? Like you said, there was a apical and a basal layer, or was it the same cell
No,
from
so
the
they're
top
not.
and the bottom?
It's the same cell. It's just that
Okay.
we were trying to find out where the orientation was emerging
Hmm.
because it wasn't making sense to me when we were looking at the orientations of the cells just on the apical surface.
Hmm.
It seemed it wasn't correlating with the elongation. It wasn't until that I looked further in and saw the basal side was actually highly aligned, where the apical surface was uh, the cells were more disordered in kind of their shapes.
Right. So I guess I'm missing something here, because how do you align the bottom of the cell and keep the top unaligned? Unless
I
I'm
mean, you have
misunderstanding this.
because you're doing this by different means. So there is alignment on the top, but it's not necessarily. But you'll have more when you're looking at you'll have more like multicellular alignment. So right, so you might have a squamous cell, but you would have alignment in terms of cell junctions over multiple cells. Does that make
Hmm.
sense?
Can
So you
you,
could have square cells, right? But they were all aligned perpendicular, right? If you look down one. But here we find actually elongated cells. So it's. Do you want me to show you an example or
yeah, if you can, it's still not clear in my mind.
right? Maybe I just go back. Uh, so if you look at um, so if you look at the cells, so I've numbered some cells on the top that are they're all the same cells. You have one, two, three, and four, and you see on that basal surface on the first example, they're quite aligned. You can see that they're sort of stretching, pulling out, right? And on the top, it seems a bit disordered. But if you go down into, as you get this contraction, the
Do you
lines you
mind,
can
sorry, do you mind
begin.
just using your pointer so I can
The
see what you're
pointer, my
talking?
pointer's not working. I'm really sorry.
Okay, so okay.
I couldn't get it to. Yeah, I would have loved to, but yeah, I
So we're
suddenly
looking at,
realized.
so which one are we
So
looking?
So now, so these are all the same cells. So I've numbered one, two, three, and four.
So we're looking at the top images, the top
Yeah. So if
three.
you're looking at the top one, you see the apical surface cells are quite disordered, or they're even elongated in the in the other direction. And if you go down to the lower picture now, you
Yeah.
might be able to see, if you look at 1, 2, 3, 4, 5, you see the junctions on the outer boundaries are aligned, but the cells themselves aren't really elongated in the same way. But you have
Okay.
these lines of tension that run through multiple cells
Okay.
of the junction. So that's what I was trying to say about cell shape, and the difference between having sort of, junction tension as compared to basolateral sort of pulling on your neighbors through, if that
Cool.
makes
Thank you. Yeah, that makes
no
sense.
worries.
Thank you. One last follow-up question, if no one else has a question. Is there a paper out on this work that we can read? I'm particularly interested in, you mentioned that there's a crawling mechanism that underpins this process, and you looked at the molecular pathways that are involved. Can you share a paper with us that we can read on this? Or
Yeah,
maybe your
sure.
paper?
This paper is out, that has more on the crawling. So there's a whole computational part that I haven't put in this presentation. So yeah, and then from there's a whole bunch of papers from the Wallingford lab that looks at specifically these crawling mechanisms. And yeah, I can send you a couple of links for the most prominent ones.
Yeah, thank you.